Okay, class, we are talking about transfer of lipids. So the PowerPoint that you're looking at is serum lipid transport. Serum lipid transport. To set the stage, lipids need to travel through the bloodstream. The bloodstream is primarily water. So there's going to be trouble there because lipids are water insoluble. So there has to be some fancy method in order for an insoluble substance to travel in the blood, in the water. And that's what this PowerPoint is about. Okay. In this PowerPoint, we'll talk about various lipids. Okay. Here is the recommended daily diet. I'm not going to get into that. This just shows the breakdown ideally. Some of you will have to take a nutrition course, so you'll have that, and then they'll expound even further. Cholesterol is a major thing. Cholesterol comes from your diet, we'll talk about, but as animals, you also can synthesize it. Animals can also make cholesterol. Okay? All right? So here's daily recommend for fats and so forth like that. Again, you can glance at it at your leisure, but it's not something I'm going to concentrate on in this particular section. We always hear about cholesterol being bad, but here are some major functions for cholesterol. So you can see you would die without cholesterol. It's just the fact that we oftentimes have too much cholesterol. So here are some functions that you can look at. Some of them you saw in ANP1, like part of the cell membrane and things of this nature. Cholesterol also acts as the precursor molecule for the synthesis of vitamin D and all your steroid hormones, which would be like testosterone, estrogens, things of this nature there. Here, right here, is the structure of cholesterol, another ANP1 thing. All steroids have 4 rings. They call it a cyclopentaphenethrin ring structure. Don't worry about that big word. But all steroids have this as the... So what's the difference when steroids? Go back into my ANP1 PowerPoints if you'd like, is where the side groups are and where the double bonds. Now, cholesterol is a sterol. You say, what's a sterol? A sterol is a subtype of steroid, All sterols have a hydroxyl group at the 3rd carbon. So cholesterol is a sterol, is also a steroid, but it's a sterol type. Okay, go a little further. What's the cholesterol ester? You may see when cholesterol is being carried, the cholesterol will hook to a fatty acid. So here's the cholesterol here, but it will hook to a fatty acid when it's being carried. It will hook to a fatty acid. Okay, that's the fatty acid right there, symbolized as an aura group. Aura in organic chemistry is like X in math. All right. Here's some dietary sources for cholesterol, which many of you probably already know. Okay. However, as animals, we also make it in our bodies. And of course, you know, humans are a type of animal. Okay. Now, the conversion starts out with something that doesn't even look like a steroid. And then there are several steps, several enzymatic steps in order to make cholesterol. Several enzymatic steps in several enzymes. But the main enzyme, the one that has the main enzyme, the bottleneck, shall we say, is this enzyme, and I want you to know this one, called HMG-CoA reductase. HMG-CoA reductase is an important enzyme in cholesterol production. It's a bottleneck. So, in other words, if you block this right here with medication, you would reduce your internal production of cholesterol. You would produce your internal production of cholesterol. So individuals that have high cholesterol in their blood, and we've given them a diet and they're compliant, that means they do what they say, and after reducing their cholesterol intake, they still have too much, then that means obviously they're making too much. So then we will give them medications that will block some of this enzyme. Not all of it, because then they wouldn't have no cholesterol. But the medication will block some of it, then which would decrease the internal production of their cholesterol. So again, a major enzyme. That's where the statin drugs, like Lipitor, you'll hear about work. All right? So this is where we talk about it. Okay. I'm going to go now to So what carries lipids in the blood are something called the lipoproteins. What carries lipids in the blood are something called lipoproteins. And what a lipoprotein is a molecule that's part lipid and part protein. A molecule that's part lipid and part protein. Okay. So we come here. So serum lipid transport. Okay. Now let's go back. Some lipids you bring in from your diet, as we talked about, and some lipids you produce, we just said, internally. Here's a list of lipids that you have. This is from ANP1. Okay. Serum lipids involved in metabolism that need assistance with transport. These are the ones that would need assistance with transport. They are lipids, fatty acids, triglycerols, cholesterol, your fat-soluble vitamins. Remember what your fat-soluble vitamins is? Hey, let's put those there. Your fat A, D, E, and finally K that's been trying to creep in. So A, D, and K are your fat-soluble vitamins. Okay. Now, here is the general lipoprotein structure. I'll pick one out over here just to give you. Here's the general lipoprotein structure. Okay. Proteins can like water and hate water. So proteins can be what we call amphipathic. Amphipathic, I should say, is where a molecule can like water and yet hate water. So the general lipoprotein structure is that the proteins are on the outside. That's what sees the water. So there would be the circulation. On the inside, you place the lipids. On the outside, the protein, and on the inside, the lipids. That's kind of the lipoprotein structure. See what I'm saying? Okay. So surrounding the core, amphipathic and hydrophilic substances and things of this nature. The density of the lipoprotein depends on the relative percentage of the protein. The more protein, the more dense. The more protein, the more dense. And we'll be seeing that. But this is the general structure. Protein on the outside, lipids kind of tucked in. Okay? So we'll go further. Okay. The first thing we get to in this whole thing is something called a micelle. Okay, so I'm going to come here. The first thing is something called a micelle. Now let me set the stage for this. When you eat lipid, so let's think about us eating fat. The fat would be eaten, taken through the stomach, and finally get into the small intestine where we absorb. What do we do with absorption? Take our nutrients that we brought in and have metabolized and put them in the bloodstream or the lymphatics. In the bloodstream or the lymphatics. Now, for lipids, remember, they first go through the lymphatics. Go back and study that we said. All right. But the problem is the inside of the intestine has a water layer. The inside of the intestine has a water layer. So your lipids now would float on top of that water because lipids would not go underneath the water layer in your small intestine because how could they? They're lipid soluble and you got that water. So in order to make the lipids soluble in water, the job is called emulsification. The process involved making lipids soluble in water called emulsification. We have to form something called a micelle. The micelle's job is to try to emulsify the fat, so let's say it'll slip underneath the water layer to make it simple. Okay? Need to get lipids underneath the water layer. All right. What happens is to do that is, I'll come here, your liver makes a substance called bile. It trickles down this duct and is stored in your gallbladder. So the liver makes bile and the bile is stored in the gallbladder. The liver's still making it, but it's stored in the gallbladder. Now, the gallbladder and the lipid and the liver have a duct ultimately that will lead into the small intestine. See, here's the lumen of the small intestine. A duct that will lead into the lumen. So bile made in the liver and stored in the gallbladder can trickle down and go into the small intestine when fat is there. A hormone is produced to make the gallbladder contract called cholecystokinin, but that's unimportant to you at this point. But remember, bile is made here. Now, we go to what is the function of bile then. Bile's job is to do the emulsification. Bile's job is to do the emulsification. Here's the composition of bile. Within the bile are these red things, the things I highlighted in red. These things I highlighted in red are the things that do the job to make the micelle. Okay? So the micelle is not a lipoprotein. It's not a lipoprotein. Here's the micelle. What the micelle consists of are these things here on the outside. And in the middle here would be lipids in the middle. And this stuff here would be on the outside, including phospholipids. So the lipids is in here, and this right here is more of a water soluble made by these things here in red. Now what that does then is allow the micelle, once it's there, can slip underneath the water layer and go to the top of the intestinal epithelial cells. Slip under the water layer and go to the top of the intestinal epithelial cells so you can start absorption. Here would be that right here. Here's kind of a micelle. Here's the intestinal epithelial cell, and it just kind of goes and helps bring the stuff in. Kind of goes and helps bring the stuff in and everything. We'll say we're not going to get real fancy on it. So the micelle's job is to get it to the top of the intestinal epithelial cell. Okay? Now, so that's as far as we've gotten. So right now, with the micelle, it's gotten it in through underneath the water layer and gotten it to the top and into the intestinal epithelial cell. All right? So that's where we are right now. Okay? So then we go a little further. All right. So now, within the intestinal epithelial cell, the next job is to take it through the intestinal epithelial cell. Take it through the intestinal epithelial cell. So now we're getting ready to come into the first lipoprotein. The first lipoprotein. So I'm going to end right here and start another one. So right now we're inside the intestinal epithelial cell. All right, let's stop here.